Okay, so we went through basics of enzymes, energy transfer, chemical reactions, how enzymes are regulated, and somewhat of that is to now to look at how cells convert some nutrient molecules into a form of energy that they can use because cells don't use carbohydrates directly for energy. It's what you find out. Oh, you need lipids or carbohydrates or things for energy. Well, you need them to be converted into a form that the cell can use for energy, but it's not those molecules themselves, right? And these molecules are not converted into ATP. So the next thing you kind of get is that, oh, well, the cells use ATP, so carbohydrates are turned into ATP. That doesn't happen. There's no such thing. Energy from carbohydrates is stripped away and then given to other molecules. Those actually through a whole series of processes. Then they give it to something else, and they give it to something else, and then eventually there's a process that uses that energy to make ATP. So the cells will use energy from the carbohydrates to make ATP molecules. Sometimes it's kind of direct, but other times it's mostly through a series of steps. And that's what we're going to start right now is some of those steps and some of the basic terms and the concepts that go on. Okay. So the idea here is that the carbohydrate we're going to focus on in the process called glycolysis is glucose, which has a chemical formula of C6H12O6. That's a glucose molecule. What is going to happen ultimately through the series of reactions that we will go through? It looks a little funny because I did this whole lecture already and there was no sound. So I'm redoing it and I wrote stuff and I had to re-erase it. So it looks a little funny because so we're going to look at glycolysis, pyruvate oxidation, and the citric acid cycle. These are a series of processes that are going to be essentially stripping the hydrogens away, separating them into electrons and protons, and then kicking out carbon dioxide as waste. Carbon dioxide, so the 6 carbons here are going to be kicked out as 6 carbon dioxide molecules as waste molecules. So it's obvious, right? The carbons they're not important. They're not being used here specifically for energy. The oxygens, we end up with more, right? There's 6 here. That's 6 times 2, so that's 12. There's actually oxygen added into this process. What was removed? The hydrogens. So the energy that we're going to use to make the ATP molecules, the energy coming from the sugar is actually coming from the hydrogens of the sugar. And then even more specifically, it's going to be coming from the electrons from those hydrogens. So it's why the chemistry part of the class, kind of right at the very beginning, is really important because it connects, to these other things. So this whole process is really linked to an oxidation, a whole bunch of oxidation events where electrons have been removed from one thing and then given to something else. So understanding a little bit about what they are, about the individual elements, is really helpful in understanding now more about this process. So let's talk about glycolysis for a second. The name glyco means sugar. Lysis means to break. And so that's part of what's going to happen in this process. The 6 carbon sugar is going to be broken in half. And so in glycolysis, through a series of steps, we're going to end up with 2 3 carbon molecules called pyruvate. We're going to put two molecules called pyruvate. So all 6 carbons will still be here at the end of this process. So they're not going anywhere yet. There's no carbon dioxide made during this process. Glycolysis will happen with or without oxygen. So if there's oxygen present, this will happen. If there's no oxygen whatsoever, an anaerobic environment, this will still just happen. Okay. For aerobic respiration, if oxygen is present, then the pyruvate will go on into the mitochondria in eukaryotic cells. So animals, plants, fungi, protists, it'll move into a mitochondria, and then the rest of it will continue. In bacteria, all the stuff happens in the cytoplasm, so there's no mitochondria in bacteria cells. But they still do the same reactions. They still carry them out in pretty much the exact same way. So glycolysis happens with or without oxygen. It happens in all cells, bacterial cells, animal cells, plant cells, archaea cells. That's why you'll see it in many courses. You'll see it because it's a common process for all living things. So everything that's alive does glycolysis. This process and these other processes. So take note of this for this particular lecture and the next couple lectures. If you're a business major taking this class for your science elective, or an art or history or some other sort of non-science major, this is going to be all you need and more probably about these processes. If you're taking this class, even though it's a non-majors class, as a prerequisite for another class like microbiology, this is not going to, what I'm going to cover is not going to be enough here. I have covered those details in other lectures that are highly detailed and more detailed on glycolysis, pyruvate oxidation, electron transport chain, all the things we're going to cover. I cover again in additional lectures that you can view as an option if you're going to need this course as a prerequisite for something else specifically. If you don't, you can still watch them to get more information, but that's not going to be required of you for this particular course. So I just want to make it clear because the objectives of this course sometimes have been very blurry for people. It's a non-majors biology course for people like business majors and others. When people are advised to take it as a prerequisite for other biology courses, they sometimes may not get enough detail, and then they do poorly in those classes. You're not going to do well in microbiology if you only get the little bit I'm going to talk about here. I would expect a lot more for those students to know a lot more than what I'm going to cover right now, because I cover that in the majors biology, cell biology class. So just keep that in mind. This is all you need for this course, this semester. But if you're going to take other classes, I have additional detailed information that you can also view. So this process of glycolysis breaks down the sugar into 2 pyruvates. It's a process that requires some input, so it needs 2 ATP molecules to come in. So it has a cost to it. It doesn't just occur without anything. So if you don't have ATP, you can't even make ATP in this particular case, which is kind of seems weird, but that's just the way it is. It also is going to use another molecule that I'm going to point out here for a specific reason, because it's going to keep coming back. It's called NAD+, okay. And we need 2 of these, okay, for glycolysis as well. And this is related to fermentation. It's related to other sorts of things you may get into in other classes, but it's also going to be what we're going to follow through all the processes. And it's what we're going to follow once these processes are done, and we still essentially have not made hardly any ATP at all. You're going to wonder, well, where's all the ATP? Well, we haven't made, these processes don't really make much of it at all. But they are going to make a lot of something from this, and that's where all the energy is going to be. So I'm pointing it out because this is going to be our real focus for the process. In the end, as these things are used up and broken down and these 10 reactions of glycolysis kind of occur, we're going to make these 2 pyruvate molecules. We're also going to make 4 ATP molecules. And then we're going to make 2 of these things called NADH. So it's where we're going to add something to that. So this is kind of what glycolysis does in a super simplistic nutshell. It takes a glucose molecule, an ATP, and this thing called NAD+, which is an electron, E with a little dash, electron carrier. It takes that and then makes 2 3-carbon molecules called pyruvate. I'm not going to draw the structure. There's 3 carbons, plus there's hydrogens and oxygens attached to it. It makes 4 ATP. So the 2 ATP you used kind of get replenished, and then you get 2 extra. So you do actually gain. So you don't gain 4, but you gain 2 ATP in this process, right? So it's like buying a lottery ticket for $2, winning 4. Technically, you gained 2, and that's not much. But if every single time you bought a lottery ticket, you were guaranteed to win $2 and you just did this a million times, you would have $2 million because you get 2 each time. And that's what glycolysis is like. It just keeps churning and going and going and going, and it doesn't make much ATP for the cell, but it makes something for the cell. And it happens with or without oxygen, and it can keep making ATP just a little bit. But most of the energy is going to be locked up here in this NADH molecule. One NADH is 3 ATP. In terms of an energy equivalent, it's worth that much. It's worth 3 of them. So that's really going to be the focus for the future of what we're going to do. What happens in glycolysis, not going to go into all the details. We don't necessarily need it for this class. You can see it again in the other lecture, and you can look a little bit in your book, but there's going to be a couple points I want to fill you in on. It's not all the chemical reactions, but what's going to happen is you're going to get this 6 carbon glucose. Okay, 1, 2, 3, 4, 5, 6. You're going to get the ATPs, and I'm skipping all the details of these particular steps, but I'm going to just plug in some of these key important parts. A phosphate from one ATP is going to get attached here, so he's going to leave as ADP. And a phosphate from a second one is going to get attached here. So we're going to have a sugar with 6 carbons with a phosphate on each end. And again, a phosphate is phosphorus with 4 oxygens. That freely by itself. And then it can be attached to other things, or it could just be alone. So these are now attached here, so there would be a bond connecting it. That molecule gets split. So that's the lysis part, the breaking part, into 2 3-carbon molecules, each of them with a phosphate, right, like that. So this is now times 2. So there's 2 of them. And there's more to this that gets rearranged and all, but this is the key molecule here. What then happens is we get some free phosphates. Free meaning they're not attached to anything. They're not attached to ATP or ADP or any other molecule. They're just free. And they're going to be attached here, but that's going to cost energy. So what's going to occur is an oxidation event. It's where electrons from the hydrogen are going to be removed and given to that carrier. So given the electrons to NAD+, and that's what we're going to make the NADH. But that process is going to store energy. In addition to storing energy here, it releases enough energy during the process to add this extra phosphate. So we end up with a sugar, well, piece of a sugar, half of a sugar molecule that then has these 2 phosphates attached to it. The next reactions strip off these phosphates. Okay, so that gets stripped off, given to ADP to make ATP. And then this one gets stripped off, given to an ADP to make ATP. That happens twice, right? Because there's 2 of these. That's times 2, and that's times 2. So that means glycolysis will make 4 ATP molecules. The reason you're able to gain something is because of this step here, right? It's the step where the free phosphate comes in due to the oxidation event that removed electrons, because it provided energy for it. If we had to use more ATP just to attach those, it means we would say use 4 and then gain 4. But that means we gain actually 0. We don't gain any, because we have to gain some. We can't use ATP to make everything that we have here. So these are additional ATP that are made from other energetic pathways, okay, other than directly using ATP just to make them. So 2 of the ATP are invested. Energy from them are used to kind of make more ATP, but they cancel each other out. The free phosphates that come in, they're new, so they're gained by the process. So glycolysis can gain a little bit of ATP to new ATP that didn't exist before through an oxidation event that's going to release energy. At the end of this process, I'm going to end this here because we run out of space and all. We're going to have this just a 3-carbon molecule. So I'm going to erase everything else kind of but that right now. All right, it's okay. It's going to be a pyruvate molecule, so you're going to see. So there's going to be hydrogens, carbons, oxygens attached to it. So don't worry about its structure. Okay? Don't worry about the structure of this. It's just going to be a 3-carbon molecule called pyruvate. And if there's oxygen present, it will be transported then into the mitochondria. And then that's where the next steps will continue. So glycolysis only happens out here in the cytoplasm. And for all cells, it'll be in the cytoplasm. So for bacteria, plants, animals, everything in the cytoplasm. The other reactions will still happen in the cytoplasm for bacteria, but for our cells and for all eukaryotic cells, the pyruvate has to go into the mitochondria to continue. It'll only go in if oxygen is present. And we call that aerobic respiration, which is requiring oxygen. So if oxygen is present, we'll move on to the next set of reactions, which are going to involve essentially breaking this down, all 3 carbon into individual carbon dioxides through 2 more processes, pyruvate oxidation and citric acid cycle. Right? So that's what we would do next.